Welcome you all. We are going to start our technical session 3 within a couple of minutes. And a senior principal scientist from National Botanical Research Institute, Lucknow, will be joining us very soon. And he will be speaking on the development, characterization, and documentation of new varieties of ornamental and innovative way. This topic is important not only for the horticulture people, but all the agriculture people, because development, characterization, and documentation of the new variety of any agricultural crop is very important, and this is the dire necessity of today's agriculture. And Dr. Roy will be explaining the different innovative way of this. So Dr. R.K. Roy is a retired senior principal scientist from NBRI, and he has recognized in many international platforms like Royal Horticulture Society London, etc. So we will go through his profile in detail after when we start this session with him. He's just going to join us in a couple of minutes. So please bear with us for 5 to 7 minutes, and I request to all the participants who have not joined yet, please inform if they are known to you, please inform them to join it. Entire schedule of our eFDP program, some of the important lectures by the important experts. So we'll be having tomorrow, we'll be having Dr. V.K. Tripathi. He is a horticulturist and head of the Department of Horticulture from CSIR University, Kanpur. And he'll be speaking on the importance of horticulture in making smart agriculture. In the evening session, that will be our technical session 5, a principal scientist from Indian Agriculture Research Institute, expert of the biofortification of cereal, Dr. Y.S. Sivai, will be here, and he will be speaking on the topic agronomic biofortification, food and nutritional security. On the day 5, in the morning session, which will be on 11 to 12, that will be our technical session 6. Director of Research, Sardar Vallabhbhai Patel, University of Agriculture and Technology, Meerut, Dr. T.P. Singh, will address on the machine for agriculture, its scope and challenges. Then another scientist from NBRI, Dr. Mridul Shukla, he will be speaking on the plantation and conservation of ecosystem. On the 12th evening session, that will be technical session 9, we will have a member from Krashak Samriddhi Ayog Uttar Pradesh, Mr. Shyam Bihari Gupta ji, and he will be talking about different innovative ways of natural farming. And he also will be talking about the FPO system in agriculture, how it is required, and he will throw a light on the government help and what is going on from the development side, from the government side for the agriculture, especially in terms of natural farming. Then on the last day, on the 13th, we will start day with the Padma Shri Bharat Bhushan Tyagi ji, who will be speaking on the खेती का सार्थक विकल्प, खेती के कुछ अच्छे विकल्प, उन पे चर्चा करेंगे पद्म श्री भारत भूषण त्यागी जी. और फिर हम लोग इस पूरे कार्यक्रम को पूरा करेंगे हमारे मुख्य अतिथि के साथ में, जो होंगे डॉक्टर प्रभात कुमार, कमिश्नर हॉर्टिकल्चर, गवर्नमेंट ऑफ इंडिया. तो पूरा का पूरा कार्यक्रम रोज दिन में 2 टेक्निकल सेशन के साथ में बना हुआ है, और एक टेक्निकल सेशन पूरी तरीके से अच्छे लोगों के द्वारा बनाया गया है. वो अपने अच्छे प्रेजेंटेशंस के साथ में आपके सामने आ रहे हैं, और जिन लोगों ने भी अटेंड किया है हमारे पहले सेशन से अभी तक के सेशन को, आप लोग निश्चित रूप से ये रियलाइज़ करेंगे कि हर एक सेशन में जो जानकारियाँ दी जा रही हैं, वो हम सब लोगों के लिए बड़ी महत्वपूर्ण हैं एंड वी कैन इनकलकेट इट एंड वी कैन डिलीवर इट टू द स्टूडेंट्स सो दैट कैपेसिटी बिल्डिंग ऑफ फैकल्टी ये हो सकता है आसानी से। तो मेरी बार-बार आप सबसे यही प्रार्थना है कि जितने भी टेक्निकल सेशन चल रहे हैं, इनका समय और इनके बारे में आपको ग्रुप पे बताया जा रहा है। आप लोग थोड़ा ग्रुप पे अलर्ट रहेंगे तो आसानी से जुड़ सकेंगे और प्रयास कर लें जितने ग्रुप में आप जुड़ पाएँगे, जितना ज़्यादा टेक्निकल सेशंस को अटेंड कर पाएँगे, उतना हम सबके लिए अच्छा होगा। क्योंकि हमारे इस पूरे एफडीपी प्रोग्राम में हम लोगों का जो विशेष रूप से सहयोग कर रहे हैं, जिसमें हमारे चेयरमैन के रूप में डॉक्टर वीरेंद्र सिंह जी, जो स्कूल ऑफ एग्रीकल्चर साइंसेज एंड इंजीनियरिंग के यहाँ आईएफटीएम यूनिवर्सिटी में डायरेक्टर हैं। उनके अलावा डॉक्टर रमेश पाल और डॉक्टर कृष्ण पाल, जो एचओडी हैं एग्रीकल्चर इंजीनियरिंग और एग्रीकल्चरल साइंसेज के रिस्पेक्टिवली हमारे एग्रीकल्चर स्कूल में। ये दोनों लोग एज अ को चेयरमैन बनकर के इस कार्यक्रम में हम लोगों का सहयोग कर रहे हैं। डॉक्टर सत्यभान सिंह ऑर्गेनाइजिंग सेक्रेटरी के रूप में इस कार्यक्रम को बनाने, चलाने और इसकी सारी की सारी व्यवस्था बनाने में हम लोगों का सहयोग कर रहे हैं। इसके अलावा कोऑर्डिनेटर्स के रूप में संकेत सावंत, अभय सैनी और अब आदित्य नारायण चौबे, डॉक्टर आदित्य नारायण चौबे जी हम लोगों के साथ में हैं, जो इसमें टेक्निकल टीम के रूप में हम लोगों के बीच में काम कर रहे हैं और जो भी टेक्निकल इश्यूज हैं, उनको देख रहे हैं। को-ऑर्गेनाइजिंग सेक्रेटरी के रूप में डॉक्टर यशपाल सिंह जी और डॉक्टर विनोद कुमार धांगर जी हम लोगों के साथ में हैं, जो इस पूरे के पूरे एफडीपी प्रोग्राम की जो बहुत सारी आवश्यकताएं हैं उनको पूरा करने में हेल्प कर रहे हैं। एक और इम्पोर्टेंट इंफॉर्मेशन है जो आप सबको देना है, वो ये कि आप लोगों ने इसके ब्रोशर को देखा होगा और ब्रोशर में ये दिया गया है कि आप सब जितने भी पार्टिसिपेंट्स हैं और हमारे जो स्पीकर्स आ रहे हैं वो भी हम लोगों के साथ में एक बुक चैप्टर शेयर कर रहे हैं। इस ईएफडीपी के बाद में एक आईएसबीएन नंबर वाली एक बुक रिलीज हो रही है। तो आप में से जो लोग भी 20 मई तक अपने चैप्टर्स हमारे साथ में शेयर करेंगे, ब्रोशर में जो थीम है इस एफडीपी प्रोग्राम की, उस एफडीपी प्रोग्राम की थीम से रिलेटेड अगर आप लोग कोई बुक चैप्टर यहाँ भेजते हैं, तो रिव्यू के बाद में हम उनको एक बुक में शामिल कर सकते हैं। सो इट्स एन गोल्डन अपॉर्चुनिटी फॉर द पार्टिसिपेंट्स to and I would like that you people fetch it and get a book chapter published in the upcoming book with the ISBN number. So again, you people are very welcome in this FDP program. Very shortly we will start our this session. joined with today's speaker, Dr. R.K. Roy, and he is a retired senior principal scientist from NBRI Lucknow. And today he's in NBRI. He was there since morning for official work. So we'll start with you, sir. And thank you very much, sir, for joining us. And in spite of your very busy schedule, for accepting our invitation for your talk. Thank you very much. And I would like to introduce to today's speaker to the audience. Dr. Roy is a person who has dedicated his life for the improvement of floricultural and ornamental plants. Dr. Roy is a renowned horticulturist in India, and he has worked in reputed research institutes like CSIR-NBRI, National Botanical Research Institute, Lucknow, and Agricultural and Horticulture Societies of India, like Kolkata, in various capacities. Dr. R.K. Roy is a former senior principal scientist and head Botanic Garden, Floriculture and Landscaping Division, NBRI, Lucknow. 1989 to 2016, he has worked in for the Botanical Garden in the NBRI. Prior to that, Dr. Roy was Assistant Secretary and Horticulturist at the Agriculture Horticulture Society of India, Alipur, Kolkata. His main field of specialization is ornamental horticulture, floriculture, and landscaping. During the tenure of his service, over 35 years, Dr. Roy has been deeply involved in the research and development work on floriculture and landscaping. Dr. Roy is member of various professional and academic societies of India and abroad. He is Senior Vice President of Bougainvillea Society of India and former Vice Chairman, Commission, Landscape and Urban Horticulture, International Society of Horticulture Science, Belgium. He was also awarded with international fellowship, scholarship, like a Royal RHS Financial Award by Royal Horticulture Society, London, UK, in 1998, Commonwealth Science Council Fellowship, Commonwealth Science Council, United Kingdom in 2003, Koch Trust Award by Royal Horticulture Society, UK. Moreover, Dr. Roy had international exposure by way of visiting many countries and a leading botanical ornamental garden in United Kingdom, France, Poland, Switzerland, Germany, the Netherlands, etc. He has written many books like Ornamental Annuals, Houseplants, Kitchen Garden, Home Garden, Gladiolus, Gerbera, Marigold, Fundamentals of Garden Designing, and Ornamental Trees of India, etc. After retirement, Dr. Roy is the founder of Roy's Green and Garden Foundation. And this is a foundation situated in Lucknow. It is working for the development of ornamental horticulture, not only Uttar Pradesh, but for entire country. People are coming, students, faculty members, some growers, and some urban landscapers, consultant. They are coming in their office, in their foundation, to learn about the floriculture and landscape techniques. Dr. Roy's contribution made a significant breakthrough in the diversification of agriculture. I would like to mention here that RGGF is organizing many internship programs, especially for the students of graduation and post-graduation. They are running for 1 week program as a 5 days program, and they are teaching them totally practical based information like how to prepare bonsai, how to management the indoor landscaping, and many more. We will later on discuss on this topic. So this is a really great pleasure for us to have you, sir, here. We are really honored and privileged and pleased to have a person like you with us in this faculty development program. And now, without taking much time, I would request you to kindly address our faculty members. Here we have got faculty members from all over India in the faculty development program. And this faculty development program is entitled with the inclusion of innovative research in education for smart agriculture. We came up with the idea that faculty should be equipped with the updated knowledge of the research so that they can deliver the right knowledge at the right time to the students, so that the student come up with the knowledge so that they can face the challenges that is coming in the way of agriculture. So over to you, sir. This virtual platform is all yours, and thank you for coming. Over to you, sir. Muted, sir. You are muted, sir. Please unmute you. Yes, sir.
Himanshu, can you please give me the title of the presentation? What I have communicated.
Yes, sir. You will be speaking on the development, improve.
New varieties.
Yeah, sir. It's a development, characterization, and documentation of new varieties of ornamental and innovative way.
Okay. Thank you. I begin with the term ornamental horticulture. As you know, the ornamental horticulture includes at present many things, not only plants and its application and its entrepreneurship, environmental improvement all together. Previously, we thought ornamental horticulture is an academic exercise only for course curriculum activities as well as for study purpose. But at present, considering the need and the changing scenario of the present environmental condition, the concept, the definition, and the purpose of ornamental horticulture is entirely different as it was 5 to 7 years back. Okay? Now, under the ornamental horticulture comes the new variety of ornamentals. Any ornamental plant you imagine, rose, the most popular flower, it is known as queen of flowers, and any other ornamental plant flowering or foliage. You will see a huge collection will coming into your mind. Okay? Then these varieties under the different ornamental crop are vast, huge, hundreds, thousands, and millions. Who cannot imagine world over the scale of magnification of new variety under different ornamentals. If you consider the commercial aspects, rose, gerbera, entire annual flowering seasonal plants, tuberose, many other tropical as well as temperate ornamental plants are there, under which several thousands, millions of varieties are running as a commercial entity all over the world. Now, the challenges and the target of the plant breeders is to develop novelty, new variety under each ornamental crop and to commercialize them. Suppose we are very popular, easily know the color of rose, red, color of gardenia, white, color of champa, yellow. But these standard colors are old concept now. The new concept is how you can change this traditional variety into its newer form, newer color, newer shape, newer structure, newer longevity. All these parameters has to be added with the present variety. Therefore, the task and the challenges of the plant breeder ahead is huge. Because until and unless they are going to fulfill all these parameters into one variety, it will not be successful as a commercial entity. Therefore, development of new variety with innovative traits, parameters is most important at the present day plant breeding program. Now comes the how you can develop new varieties. whether for academic purpose and whether for commercial purpose or for applicability as a nurseryman. There are 4 standard ways. First one is hybridization. Hybrid banana. We usually say, ये तो hybrid variety है. This is hybrid variety. Very common term, hybrid variety. And the technique is also very old. Combining, crossing 2 desirable parents, male and female, which are having desired characters as per your breeding targets. Now you have to consider before taking up hybridization is that what is your target? First to decide your target, that is color, fragrance, flower size, number of flowers per branch, stem length, etc. etc. It could be a dozen of characters, which could be a breeding target for a plant breeder for development of new varieties. So all these characters has to be limited into 3-4, not that you can achieve 12 parameters in one variety, not possible. You have to minimize 2-3 parameters, either color, fragrance, flower size, number of flowers as the most. Then you select accordingly the parents, male parents and female parents. And obviously, both these parents must have the characters you have decided as a breeding program, in the breeding program as a target. So, selection of parents is another important character when you going for hybridization. And development of new variety is not a easy task, time taking. Depending upon the plant species, it may take 6 to 8 years. 6 to 8 years, depending upon the plant variety. Suppose what I did, I developed, say, about half a dozen variety in gladiolus. I give a classic example out of my experience. See, it took generally 6 to 8 years to develop a new variety of gladiolus. And the procedure of hybridization is easy, but you get only a few seeds like mustard seed. Then you sow them, you get some bolder seeds in the form of a pea. Next year, again you sow them, you get a bigger size, double size of the peanut. Then next year, you can have small bulblets which could be able to produce flowers to see the flower color, etc. And then again selecting those progenies out of the flowering plant, selecting them again and multiplying them again. So it takes about 6 to 8 years in case of gladiolus. On the other hand, if you take vegetatively propagated plants like stem cuttings, rhizomes, bulbs, tubers as a breeding material, then there is another method which is known as gamma radiation. In gamma radiation, it's a technique, a machine which releases gamma radiation in a definite dose prefixed with the crop. You have to research which dose, how much time of exposure you have to give for each and every crop. It is different. Say for canna, I did on canna gamma radiation, rhizomes cut into pieces then put in a container, put inside the gamma chamber, expose the cut pieces of rhizome into different exposure with short duration, medium duration, long duration. Then you sow them, rather plant them into soil manure mixture bed or in pots for sprouting and growing. After growth, when they flower, you see whether there is any change from the parent or not. If there is change, whether that is desirable or not desirable. Something undesirable characters also phenotypically expressed, which is not at all important from the commercial point of view, then you have to reject those progenies. Out of 100 progenies, you have to select the best one and as per your breeding target also. Then you again go for multiplication of the selected progeny into a mother plant, from mother plant again into daughter plants by vegetative propagation, tissue culture, etc., whatever facilities available for mass multiplication. So far I have described two methods of development of new varieties. First one was hybridization and second one is gamma radiation. Gamma radiation advantage is, since vegetatively propagated material can be irradiated like rhizomes, bulbs, seeds, so quick result is got. Suppose canna, if you treat rhizomes in a definite exposure and plant them in pots, within a year the plants grow and the next year it flowers. Once it is flowers, then you select whether it is good or bad. If it is good, then you treat that plant progeny as a mother plant and again propagate from mother plant to progenies. So it takes about 3-4 years. In comparison to hybridization, the time requirement is half. 3 to 4 years you can get results. So this is something advantageous over the hybridization. But hybridization has got other advantage, thorough change of genetic basis among the progenies and very stable one. Very stable one, and the process is little long, but you can have the opportunity to get maximum of morphological variation, desirable traits in the form of progenies. Then there are two other methods. One is chemical treatment, another is natural bird sport. Chemical treatment, it is another very easy method for the beginners, even nurserymen, hobbyists, house owners, home gardeners can practice this kind of variety development program through chemical mutagens. The mutagen is, chemical name is colchicine. It's a powder form chemicals which can be diluted in a definite ppm 1500, 2000 as per requirement of the plants, and then you have to apply in a definite way. I will just explain you how this could be applied on the plants. For colchicine treatment, you have to select the mother plant of different genus. Suppose you want to do it in hibiscus, you want to do it on bougainvillea, canna, many other plants, even on house plants also, even any kind of flowering plants also. Select a healthy mother plant. The mother plant should be placed in a very congenial microclimatic condition required for growth. Not that you have kept the mother plant in full sun, which is adverse to the growth, not under full shade, which is again not congenial for growth, and the soil mixture, etc., should be very right so that it grows first and the plant growth is healthy. After selection of a mother plant, you have to select the branches which are growing in a healthy way. The apical bud, the apical portion of the plants is very healthy for application of the chemical. What do you have to do? You first tag out, put a label on the branch, what you have selected. B1, branch 1. Same plant, B2, branch 2. Then you again out of the branch select apical leaves, say buds, etc., which is growing at the top of the branch. Again you sublabel them, either writing on the leaves or in any kind of label so that you can identify afterwards that you have applied chemical on that part of the plant. That is the only purpose of labeling. Okay? Then you have to prepare solution of colchicine chemical in specified dose. You have to consult literature which dose is recommended. And time of application as well as frequency. Three things: concentration, then time and frequency. This differs among the ornamental plants. May be different from one and another. Hibiscus may be different, roses may be different, other plants, house plants may be in the different duration, different concentration. So we have to consult literature, decide, prepare chemicals. Then comes the application part. What is generally done? One small cotton piece is kept on the apical stem. Apical stem means the growing part of the branch. Maybe a few small leaves, leaf buds may be there. We just stretch the cotton and put them over that. Then application of that treatment through droppers or anything in required quantity, say 10 drops, 15 drops, 20 drops at a time over the cotton. The cotton should be soaked. The cotton should be properly adhered to the leaves so that for absorption, so that whatever chemical you have added to the cotton, that should be absorbed to the leaves and the leaf buds present there. This kind of situation you have to manage yourself at the time of application. Application is recommended either in the morning time, 9 to 11 am, then maybe in the afternoon, 2:30 to 4:30 pm, when the plant's in active growth phase. Not in the evening, late evening, when there is no light, or not very early morning, when there is again no sunlight. So active growth phase, growth state should be there. That means presence of light is required. According to plant, frequency may be 2, frequency may be 3, not more than 3, 4 I think. And repetition of the treatment, 1 day, 2 day, 3 day, consecutive 3 day, maybe you have to treat the plants. So this is the treatment part, application part. After that, you have to observe, observation part. Only you irrigate the plant, provide nutrient, etc. So there is no stress of growth. Okay? Then you see when the new leaves are coming out, whether any change from the mother plant. Is there any chlorophyll variegation? Is there any deformity of the leaves? Is there any size change of the leaves? That you have to observe. And after that, if you see things are desirable, then you have to propagate that branch by air layering and plant them into a growing medium so that it develop into a mother plant. This is basically the procedure and the way of doing the colchicine treatment. Once the mother plant is established, that has to be observed for another 2-3 months over the years, whether the change are permanent or disappearing, or the other branches coming out, whether they are of the similar nature or they are different again. All these we have to observe and manage so that a uniform mother plant is developed. Okay? Then if you see the all desirable changes are acceptable, then you declare this is a new variety and you go for further propagation into increase the number of progenies. So this is simply the procedure. Third procedure is colchicine treatment. This is second most easy process for the home gardeners, nurserymen, hobbyists, even for the beginners and learners. Okay. Then the last procedure is natural mutation, which is technically called as bud sport. ये भगवान का देन है. There is no human interference. Only the natural light, sunlight, creates the variability. You know, sunlight is having ultraviolet ray and other things. So these things change the genetic makeup of the plants, but not very frequently, not very easily obtained. After you are maintaining, suppose 100 of plants, you can observe only 2-3 plants which is having some kind of natural variation of leaf color, flower color change, etc., etc. But it requires experienced eye to spot them, to locate them, and to isolate them. The procedure is simple. You have to do nothing. Only sunlight will play the role. But afterwards, the process for identification, process for isolation, process for regeneration, process for multiplication of further progenies are a difficult task. One has to procedure again the standard procedure of plant propagation, assessment, isolation, propagation, etc., etc. But one has to be very careful whether the mutant isolated or detected, established is reverting to parents or stable. It should be stable. Suppose one mother plant you have got, who propagated 10 plants out of that mother plant, and all those 10 mother plants should exhibit morphologically similar vegetative and floral characters. Then only you can say the characters you have identified, isolated, detected, it's stable, and it's a new variety. So this kind of evaluation process one has to go through for development of new variety. So there are 4 procedures, methods of new variety development. One is hybridization, gamma radiation, chemical treatment with colchicine, and other last what I have explained was bud spot. ये जो 4 प्रोसीड्यूर है, ये प्रोसीड्यूर्स है well established, internationally approved, acclaimed. When you develop a variety, then everybody will ask you कि आपने कैसे बनाया है, how you have developed, which procedure, then you have to name the procedure कि आपने हाइड्रेशन किए, गामा रेडिएशन किए, if it is done through gamma radiation तो डोज क्या था, कितना फ्रीक्वेंसी था, कितना साल लगा, how it is different from the mother plant. All these things have to be documented. This is one part of lecture presentation, that how you can develop new variety. ठीक है, और न्यू वैरायटी डेवलपमेंट का बाद the next step is how you can establish your claim that you have developed a new variety. When you develop a new variety, you give publicity to the scientific community, commercial nursery, everybody, that I have developed a new variety, so and so character. So you have to first prepare yourself through characterization. कि आपने जो बनाए हैं वो how far it is different from the mother plant, both morphologically as well as जो flower part है उसमें भी जो है, ठीक है. तो morphological characterization is first requirement. Morphology includes, मैं आपको कुछ दिखाना चाहूंगा कि morphological characterization, how we can do it and what are the characters you have to take. Can you see anything, Himanshu?
Yes, sir, we can see. But if you can put it some closer, then it will be very easy to.
अभी क्या है,
अभी ठीक है सर.
अभी जो है ना इसमें जो है अच्छा नहीं ठीक है, इसमें है ग्रोथ हैबिट. फर्स्ट इज ग्रोथ हैबिट. पौधा कैसे है, इट इज इरेक्ट, ड्रूपिंग एटसेट्रा. देन द लीव्स कम्स, the shape of the leaves, okay. Then the basal portion of the leaves, leaf tip, leaf margin, leaf variegation along the middle or through the margins. So all these things you have to consider before documentation. Rather, these characters has to be documented one by one. All these are vegetative characters, branches, leaves, petioles, even thorns. Okay. Then comes flower part. How is the inflorescence? Long, short, medium, whether the flowers are growing on the terminal part or all through the inflorescence or in the leaf axils. All these things you have to consider, examine and note down. As you see here, single bracted hai, multi bracted or spines. Spines is a diagnostic character, whether spines are long, medium, short, whether they are hooked or not. All these things are you have to consider and note down. Then comes the flower tube, as you see here. Even the flower tubes, whether they are attached to the petals or not. Then the floral tube part. This is specifically for bougainvillea, but same thing is applied for all the ornamental plants. Okay. So all these characters one has to be very, very careful, examine them, document them, and then you prepare a descriptor of your new variety developed. This descriptor is the proof of your new variety, because this is the basis how you can compare with the mother plant. कि ये जो variety है, this is, there is color change from white to red or red to yellow, and the color shade is so and so as per Royal Horticultural Society's color chart. So all these documentation you have to do. ये जो documentation है, इसका क्या जरूरत है? Why we say documentation is necessary? Because it establishes your claim of new variety. आपने कह दिया हमने new variety निकाले हैं, Gladiolus new variety named as Urvashi, as I have done. People working in the same crop from other institute, they will not believe. They will say, no, it is not a new variety, it's a freak, it's not a stable genetic material. So you have to establish your own research. इसलिए आपको डॉक्यूमेंटेशन करना जरूरी है, क्योंकि ultimately all those new varieties have to be registered. Until or unless you register these new varieties, you cannot claim that it is your variety. Your in the sense, your name of the breeder, as well as name of the institute, name of the university, name of the nursery, all these things have to be registered with government agencies. In case of bougainvillea, International Registration Authority is with IARI. IARI में variety registration आप करा सकते हैं. Over and above, you can register new variety with PPV and FRA, Plant Variety Protection and Farmers Right Authority, Government of India. अलग ऑफिस है इनके, इनका वेबसाइट है, वहाँ देख सकते हो क्या रिक्वायरमेंट है, एप्लीकेशन फॉर्म है, जो कैरेक्टर्स हमने बताए हैं वही कैरेक्टर्स वही है, क्योंकि ये जो गाइडलाइंस है ये हमारा ही बना हुआ है फॉर ग्लैडियोलस बोगनविलिया कैना. तो वही कैरेक्टर्स आपको वहाँ फीड करना है. प्लांट हाइट कितना है, कितना ब्रांचेस है, लीफ कलर कितना कैसे है, फ्लावर कलर क्या है, फ्लावर का मेजरमेंट क्या है, कौन सा वैरायटी प्रोसीड्योर आपने डेवलप किया, वैरायटी हाइब्रिडेशन, गामा रेडिएशन एटसेट्रा एटसेट्रा, ये सारे डिटेल्स आपको वहाँ भरने पड़ेगा, फॉर्म भरने पड़ेगा, फीस जमा करने पड़ेगी. देन they will accept your application only. Not granted that this is a new variety developed by Himanshu Trivedi. No. Then what they will do? They will, and you have to submit your breeding material there. Suppose develop canna variety, to canna variety ka rhizome plants you have to deposit to the PPBRA of authority. Then they will send those material to the testing center. टेस्टिंग सेंटर कहाँ है? IRI में आपको पौधे बोगनविलिया जमा करने पड़ेगा. Then they will send to NBRI for testing. कैना वैरायटी आप सबमिट करो PPBR में, then they will send it to NBRI. नहीं तो IIHR Bangalore. Certain center are already identified. They will then send these varieties for evaluation. Evaluate करेंगे. क्या evaluate करेंगे? Those variety documentation आपने जो बना के जमा किया है. आपने लिखा flower color is light pinkish. They will match with the flower color. whether it is light pinkish or yellow. If it is yellow, then they will comment that variety submitted was flower color was so and so, but ultimately it is yellow, not accepted. If it is pinkish, then they will say yes, accepted. If everything goes well, then they will give a good report to PPBRA कि जो variety that claimed by Himanshu Trivedi was found correct and everything was as per the documentation data. Then PPBRA will issue a certificate in the name of the applicant that we have received the application from so and so, and we found that the variety is a new one, and number is given. New number का एक ISSN number, जैसे जर्नल का ISSN number होता है, वैसे ही variety identification number देते हैं. उसके बाद एक सर्टिफिकेट भी देते हैं, और तब जाके आपका ये प्रोसेस कंप्लीट हुआ. So from breeding to registration, it's a long battle. मतलब आदमी जो है, हम रिटायर होने का पहले जो जमा किए हैं वो अभी तक NBRI में सर्टिफिकेट नहीं आया. It takes time, 2-3 years in PPBRA. Variety development 6 to 8 years, 3 to 4 years, depending upon the variety. So it's a very long process. One has to be very, very patient. Patience नहीं है, breeding program में मत जाओ. ये कोई वो नहीं है, it's not a computer game कि आपने कुछ डाला और तुरंत निकल आया. तो इसमें जो है every step to be counted, every time you have devoted to be counted, and every effort you have put in, that has to be credited into your account. When only you get the registration certificate, your journey will be successful then. But in the meantime, you will get full satisfaction and pleasure that you have developed something. It is under consideration by Government of India. That is a full credit to you and your department. This is all from my side.
Thank you very much, sir. It was very extemporaneous and very informative, and definitely blended with your vast experience of varieties of development, characterization, and registration. Thank you very much, sir. Thanks a lot for giving us your precious time. Now I request one of the member of our organizing committee, Engineer Sanket Sawant, to propose a vote of thanks from all of us. Please, Sanket, unmute yourself. Sanket, you're not audible.
I'm not audible yet.
You're not audible, Sanket. No. No, Sanket. Not yet audible. You can come to me. Fast.
Anyway, Himanshu, don't be worried about the vote of thanks. Rather, you can do one thing.
Yes, sir.
You can share my number, WhatsApp number. If anybody have any query, please do forward them to me. I will answer each and every one. Conveniently, not very instantly, but conveniently, I will definitely answer those queries.
Okay. Sure, sir. I will share your number and all details on the WhatsApp group we have especially made for the participants of this eFDP.
Okay.
And sir, on the behalf of the organizing committee, I'm really very grateful, and definitely for you especially, my words never be capable to express my deep sense of gratitude to you, sir. And this is really, we are really honored to have you And thank you very much for giving this time. Thank you very much,
sir. Thank you. Thank you, sir.
So, important announcement for our participants that our tomorrow's session will begin at 11 a.m. with the head of the Department of Horticulture, Dr. V.K. Tripathi, will be speaking on the importance of horticulture in making smart agriculture. So I request you to be on time on tomorrow 11 a.m. Thank you very much once again to the guest of the day, Professor Dr. R.K. Roy sir, and all the participants. Thank you very much.